I chaired a session that was entitled Immunoescape and Immunotherapy for Lymphoid Malignancies. Now, lymphoid malignancies are a very heterogeneous group of cancers, over 40 cancer types. And the goal of the session essentially was to address the issue. For the most common type of lymphoid malignancy, it's called diffuse large B-cell lymphoma. I presented evidence that tumors have adopted the genetic changes to escape surveillance by the immune system. They delete or inactivate genes that produce molecules that are seen by T lymphocytes in order to kill tumor cells. These are irreversible changes because they are genetics, and so over 50% of these tumors are invisible to the immune system. Okay? This is a simple, direct way for tumors to evade surveillance. In the second session, Dr. Ship from Dana-Farber showed for another type of lymphoid malignancy called Hodgkin lymphoma, a different mechanism. Cells put on the tumor cells put on their surface molecules called checkpoint that tell the T cells, the immune system, not to attack them. Okay, so although the tumor are seen by the immune system, they tell the immune system not to be attacked. The difference between the first scenario and the second scenario is that the first is irreversible. The tumor cell will be permanently invisible. In the second scenario, you can intervene therapeutically, and in fact, antibodies deactivating these so-called "don't eat me" signals by tumor cells have a dramatic effect in changing the clinical course of Hodgkin lymphoma. So that's a major advance. This type of intervention is now used for other tumors, melanoma, lung cancer, but I would say Hodgkin cancer is the most dramatic success for the time being for this intervention. It's not clear how long-term this will end up being, but certainly there's been a change in the clinical course. In the third presentation, Dr. Ron Levy from Stanford reviewed in an educational spirit for the audience all the different approaches that are currently being developed, again to either make the tumor more visible to the immune system or to tell the immune system to see the tumor better. And he presented evidence of arming T lymphocytes genetically to recognize specific antigens on the tumor, or bispecific antibodies that bring on one end the T cell close to the tumor, and when in the other domain they kill the tumor cells, and presented initial evidence on a multitude of clinical trials around the world that are developing these strategies, some with initial very good successes in B-cell malignancies. The tumor cells are making the cell invisible by putting up molecules that tell the T cell, don't attack me. Whether in the first, in diffuse large cell lymphoma, they make themselves invisible by inactivating the molecules that usually are seen by T cells. So there is a variety of mechanisms. I think inactivating the so-called checkpoints is becoming a widespread approach in many types of cancer, with, as I mentioned, success in melanoma and initial success, melanoma and lung cancer, but certainly is an approach that it is broad utilization. The first one, the simplest one, genetic inactivation molecule, is difficult, is irreversible, and probably this poses serious challenges to make the immune system more active, because the tumor cells are seen as self. Yeah, if you wish, if you rearm the immune system toward the tumors, you would mimic an autoimmune response, but for a good end in this case. There is an enormous attention to immunotherapy of cancer after decades of slow progress. I think there are three areas of major development. The first, as I said, bispecific antibodies that connect T lymphocyte, the weapons to the targets, the tumor cells, the so-called CAR T technology that also modifies T cells, the weapons to attack better. And the third is, which is the one that's attracting a lot of attention, is based on the fact that genetic mutations that make a normal cells to become a tumor generate a number of so-called new antigens, neoantigens, that are recognized as new by the immune system. And so you can use these neoantigens as vaccine for arming the immune system. This is a more complex and long term. Most cases will be different one from the other, but rationally makes a lot of sense. As a conclusion, I would say we are entering a very exciting area of immunotherapy of tumor. As I said, after decades in which this field was dormant. A lot has to do with major development in technologies that allow to modify effector cells, immune cells, and to identify new molecules that are non-self in the tumors. The technology is exploding, and following that there are new approaches every day. I think the next few years we'll see a lot of progress. This is just the infancy of this re-emerging field.